Monsieur le Président, Monsieur le Secrétaire perpétuel, chers collègues, chers amis, vous me permettrez de m'exprimer en anglais parce que le lauréat du prix Alvarenga de Piauhy 2015 est italien et ne connaît probablement que quelques rudiments de français. The members of the jury, chaired by Professor Benoît Van den Eyde and constituted by Professor Renaud Louis, Professor Dominique Bron and myself, have selected Dr. Luca Tiberi for the prize Alvarenga de Piauhy 2015. This award recognizes the highly original and significant work conducted since 2008 by Luca Tiberi at the ULB Neuroscience Institute. These studies identify the transcriptional repressor complex, which is both necessary and sufficient for neurogenesis and suppression of malignant medulloblastoma, the most prevalent brain tumor in children. This complex is a potent suppressor of the sonic hedgehog pathway in both normal and oncogenic neural development. The development of molecules able of activating this repressor complex will lead undoubtedly to promising new treatments of malignant medulloblastoma. Luca Tiberi is born in Italy on 24 February 1979. In 2004, he was graduating Master in Medical Biotechnology cum laude at the University of Bologna and PhD in Molecular Oncology in 2008 at the University of Trieste. Since 2008, he is a postdoctoral scientist in the laboratory of Pierre van der Hagen at the ULB Institute of Interdisciplinary Research and Neuroscience Institute. He has authored and co-authored 20 publications in highly ranked journals such as Cell, Cancer Cell, Neuron, Nature Neuroscience, Nature Cell Biology, and Trends in Neuroscience. On behalf of the members of the jury, I address our warmest congratulations to Dr. Luca Tiberi and wish him the best success for his future and highly promising research career. Please, Dr. Tiberi. Just a last word, Dr. Tiberi. I'm quite sure that you will get other prizes in the future because you do excellent work. And never forget that the first recognition was by the Royal Academy of Medicine.
That's true. Merci. Bonjour, merci pour le prix. J'ai travaillé 7 années avec professeur Van der Hagen, toujours en anglais. Je ne parle pas très bien français, je vais parler en anglais, pardon. During my postdoc with Professor Van der Hagen, we have studied BCL6 and its complex during medulloblastoma development. What is medulloblastoma? Medulloblastoma is the most common brain cancer in children, an infant, and is divided. It originates from the cerebellum. That's why the name is medulloblastoma. And is divided in 4 main categories, 1, 2, 3, 4, or group Wnt, sonic hedgehog, group 3 and group 4. What we know is that these 4 type of medulloblastoma are really aggressive and they are different one from another. And during my postdoc, we studied in particular sonic hedgehog, this kind of medulloblastoma. Just to give you a glimpse of what's going on in humans, what we know is that in patients that have a mutation in the sonic hedgehog pathway, they develop the tumor close to the brainstem. But what we know is that from other labs is that also in the mice, if we remove one gene, we have a genetic alteration of one gene, we develop medulloblastoma. So at that time we knew that to use mice could be a good model to study this kind of tumor also in patients. And so we can say that mouse models of sonic medulloblastoma are good as human brain tumors. Just to give you an idea what we have done, this is a mouse cerebellum. The development of the mouse cerebellum is quite simple. We have progenitors in the upper part, they will become neurons, and everything is regulated by this pathway, the sonic hedgehog pathway. And what we know also is that from these progenitors, the neurons will, the tumor will give rise. So we know that those progenitors are really important. And in fact, the granule neurons, the progenitors, are the cell of origin of this kind of tumor in humans and in mice. And just to give you an idea of the clinic, what people have done so far, they have treated patients with sonic hedgehog medulloblastoma with inhibitor of the hedgehog pathway. Here you can see this is a PET scan of a patient, pre-treatment. So before the treatment, you see that there are a lot of metastases. After treatment, there is a nice recovery, but after 3 months, there is a relapse of the disease. So that means that this kind of cancer, they acquire resistance. So the point is to find novel pathways or not other ways to target this kind of tumor, because one inhibitor is not enough. That was the aim, the goal at the beginning. To go very quickly, so just to put everything in a model, we have progenitors in the normal cerebellum. For one reason, we still don't know exactly which are all the pathways involved, they become cancer or cancer cells. We know from study from patients that there have been exome sequencing studies, so that means they have tried to find which genes are mutated in these tumors, which genes are not well modulated. And they have found that 2 proteins, BCOR and DB1, are involved in the process, are mutated. So we have studied the BCL6 pathway because those 2 are part of the BCL6 complex. So we thought at the time BCL6 could be involved in the tumor. And what we have done, we have just overexpressed BCL6. Just to go quickly, we use a mouse model where we can induce cancer. So just to make a long story short, no cancer, normal cerebellum, cancer after induction. And what we have done, we have also put a lot of BCL6 in this mouse model. So normal BCL6, a lot of BCL6. If we go directly to the results, this is a wild-type cerebellum, normal mouse, healthy. This is with the tumor. You can see marker for proliferation. The tumor is growing and the mouse is going to die. When we overexpress, we put also BCL6, we see that we have a nice reduction of the tumor. So at least we are able in vivo in mice to reduce the mass of the tumor. And here we can see the survival of the mice. This is the mouse model that we have used to induce medulloblastoma. This is the survival. As you can see, in 80 days, all the mice will die. With BCL6, we have a delay in the disease. So the disease is still there, the mice are still dying, but they need more time. So at least we have a window where we can play, a window where the mice can survive more. And we hope maybe one day we can do also the same with the patient. And just the final remarks is that I will just show you putting more BCL6 to try to stop the medulloblastoma, to stop the tumor, but to really prove that this protein in this pathway is involved in this kind of tumor, we have to remove it. So if you remove the gene and you see if you get the tumor. So what we have done, to make a long story short, we have used different transgenic mice. This is a normal mouse. This is the cerebellum of a normal mouse. You can see the cerebellum is normal, there is no tumor. If we remove BCL6, the mouse is still normal. But if we remove BCL6 and P53, it is a well-known oncosuppressor, a gene that is well-known to be involved in cancer in humans. In this case, we can see that we have a tumor. So also with this, we have proved that BCL6 is required to stop the tumor. Because if you remove it, you get a cancer. This is my last slide, where I just try to recapitulate that if you remove BCL6, you get the tumor, but you can also try to stop the tumor by putting BCL6 and to transform the cancer cells in more neuronal-like cells. This is my final sentence, where I would like to stress also thanks to Professor Van der Hagen that he helped me with all of that when I was just arrived in Brussels in 2008. I learned from him studying the cerebellum, and our message is that at the end, studying the cerebellum neurogenesis development could be also important to study novel molecular pathways that are involved in brain cancer. And I would like to thank ULB, all the people of the deviation, FNRS for the Chargé de Recherche record for 4 years, Televi that gave me my postdoctoral fellowship for 2 years, Professor Van der Hagen, all the people in the lab, in particular the postdoc Jerome that helped me with this project, all other people like Angeline, Adele, and in particular Fausto that now is working with me. I would like to thank also people of the lab of Professor Plan and all our collaborators and all of you. Thanks.